Hi, myself Chamundeshwari, Assistant Professor, Department of Biomedical Engineering at Sri Ramakrishna Engineering College, Coimbatore, Tamil Nadu, India. Today I am going to present a topic on diagnostic studies of nervous system disorders. Now, before going to the diagnostic studies of nervous system disorders, we will see what is about nervous system disorders, and what are the common causes and symptoms of the nervous system disorders, and then into the common procedures of the diagnostic procedures, and then deeply into the diagnostic studies of the nervous system. Next, we will see, first of all we will see what is the nervous system. The nervous system is a highly complex organ that controls and coordinates all body functions, especially through the electrical and chemical signaling. The cells consist of specialized cells called neurons and supporting cells called neuroglia. So, what are the common functions of the nervous system is it's split up into three: sensory functions, integrative function and motor function. Now, sensory function, as we all know, deals with the senses, that is like what is the input and the output, that is the internal external exchanges, and the integrative function is nothing but is the process and interpret information, that is the cognitive portion of the thing, that is the integrative function. And then the motor function. Motor function is usually, as the name indicates, it deals with the muscles, that is the how the muscle contractions and the gland secretions responds by the nervous system. Next, nervous system is divided into two components, that is the central nervous system and the peripheral nervous system. Now, central nervous system consists of two, that is it deals with brain and the spinal cord, whereas peripheral nervous system, it deals with the as the name indicates, the peripheral is the lateral thing. So, it deals with the cranial nerves and the spinal nerves and also the autonomic nervous system. This is all about the introduction of the nervous system, and then we have the nervous system disorder. So, any disorders that happens at the nervous system, that is the parts of the nervous system, that is the brain, spinal cord, and the nerves related associated with the nervous system, we will have a dysfunction in the nervous system. So, that is called as the nervous system disorder. It is a condition that affects the brain, spinal cord and the peripheral nerves, leading to the impaired functions. So, for example, we have a nervous system disorder as a neurodegenerative disorder, that is the Alzheimer's disease, Parkinson's disease. Alzheimer's disease is nothing but is loss of memory function, as we all know about that, many of the examples we know. And also Parkinson's disease also is related to the loss of cell tissues functions. So, the neurodegenerative is itself, it's a loss of tissues or cells disorder. Now, infectious, the disorders may be caused by bacterial or viral infections, that is leads to meningitis and encephalitis. That is, we have a weakness, paralysis, tremor and muscle rigidity, and this is related to sensory parts of the symptoms is that numbness, we will have numbness at our fingers and arms, and also tingling and then burning sensations in the all parts of the sensory organs. And next, cognitive symptoms is, as we told before, it's a memory loss, difficulty in the concentration, and also autonomic, it's like a physiological imbalance like BP fluctuation, irregular heartbeats. This also leads to the nervous system disorder. And others, as we all, it's a headache, dizziness and seizures are all the first symptoms of the nervous system disorders. Next, what are the causes? As we discussed before, it will lead by trauma like head injury or spinal cord injury, also leads to the nervous system disorder by infections and degeneration. Vascular is nothing but it's blood vessels, any blocks in the blood vessels leads to stroke, and bulging of blood vessels also leads to the aneurysm, that is nothing but aneurysm. And autoimmune, it's, as we discussed before, it's in the examples itself, it's a multiple sclerosis, nothing but is the scarring of the nerves. And metabolic causes, when it leads like diabetic neuropathy, where the diabetic patients will long term diabetic medications will lead to diabetic neuropathy. And if a person consumes alcohols and deals with heavy metals, also leads to nervous system disorder that will be toxic to the humans and then to human body, and then it leads to the nervous system disorder. Now, and also last stage is the abnormal cell growth that leads to the tumors and brain tumors. Now, next, how do we diagnose this nervous system disorders? So, that we go for the first clinical examination, then imaging studies, electrophysiology, cerebrospinal fluid analysis, blood test, and then neuropsychological test. So, these are the initial diagnostic studies of the nervous disorders. First clinical examination is like the coordinations will be checked, that is reflex coordinations will be checked in the clinical examinations, and also the basic physiological tests will be like symptoms of what are the history of symptoms and everything. It is a clinical examination, nothing but it is a physical examination of the initial procedure. And the imaging studies deals with the CT scan, MRI scan, and then fMRI. CT scan is nothing but it detects the hemorrhage, tumors and fractures, whereas MRI is nothing but magnetic resonance imaging. It gives the detailed images of brain and spinal cord, whereas fMRI measures the brain activity. Cerebrospinal fluid, it is related with the lumbar region, that is nothing but the spinal cord discs. Any infections, that is like bacterial infections or something or else multiple sclerosis will have some infections. So, we have to collect the fluid samples from the spinal cord, so that we are using this cerebrospinal fluid analysis test to identify the amount of infections and virality of the infections, bacterial infections. And then electrophysiology, that is nothing but, as the name indicates, electro means electroencephalogram is EEG, EMG is the musculogram. EEG detects the electrical activity of the brain, whereas in the cases of epilepsy EEG will be helpful, and EMG is the muscle activity test, that is how does the muscle contraction and takes place, that is related any paralysis patients for the how much is the nerves synchronization will be tested by the EMG, and followed by initially blood tests for the infections and metabolics or autoimmune conditions. And also neuropsychological test is nothing but is the cognitive evaluation in cases of dementia. Now, out of these we have a main important specific topic is that EEG. Next we are going to see about the EEG only. Specifically we will see the diagnostic procedure EEG alone. So, what is EEG? Now, as the name indicates, it means split up into three, that is electro, encephalo and graphy. Electro means it deals with the electrical activity, encephalo is the brain in Greek, and graphy is the recording. So, electroencephalogram means it is a recording of the electrical activity of the brain. So, the definition deals with, it goes like, it deals with the recording and study of electrical activity of the brain. So, now the electrodes are attached to the skull of the patients, and then the brain waves are picked up and recorded. So, brain waves is nothing but is the summation of neural depolarization. This comes associated with the senses. We have 5 senses, right? From that 5 senses we have a stimulus and also by the thought process. So, these are the neural depolarization. Now, how does this origin of EEG? Now, how does we pick up this signal? So, these signals are picked up by the graded potentials, and this graded potentials is being arises from the dendrites. Dendrites are nothing but it is a part of the neurons, and many neurons connected together which gives this graded potential. This graded potential is caused by the synaptic activity of the neurons, that is nothing but synaptic activity is nothing but is the impulse transmission between the neurons, that is at the juncture of the synapse. So, or else we can tell it as it is a junction between the 2 nerve cells, that is so at that junction the neurotransmitter at the point of the synaptic activity, neurotransmitters will be released, that is especially acetylcholine we will tell it as, and these are summed up to form the nervous tissues. Single neuron activity cannot be measured because it involves a multiple neurons to be multiple neurons. So these multiple neurons are to be synchronized, has a firing mechanisms, and our firing mechanisms are done in a synchronized activity manner, and then these synchronized activities discharge neurons and then gives as a brain waves. So these brain waves are depends upon the reticular activating surfaces. So intensity of the brain wave depends upon the reticular activating brain waves. What are the brain waves? As we discussed before, it is the intensity and patterns of the electrical activity. That is nothing but is the excitation of the brain. It is a reticular activating system, is nothing but is the mesh like neurons are in the form of a mesh like, and it's generally awakening from sleep. That is the thing, and when we are active, our brain will be active, that is the potential excitation potentials will be in the top peak. So that waves are being measured, that is called as the brain waves. The waves in the recorded electrical potentials are called as the brain waves, and the intensity of the brain waves, it ranges from 0 to 300 microvolt, and the frequencies ranges from few seconds to 50 or more than. So brain waves are irregular and there is no general pattern for it, and in the disease condition it have a distinct pattern. Like epilepsy, it has a distinct pattern. So we have 4 different types of brain waves, that is alpha, beta, theta and delta. So each one have different frequencies and different positions, different functions. Alpha waves, now alpha waves frequencies ranges between 8 to 13 hertz. It normally occurs in the normal persons. In the awake situations, when they are in a resting state, it occurs in the occipital region, and it disappears when during the sleep. So the amplitude of this wave is about 20 to 200 microvolt, and the mean of it will be around 50 microvolt. Now beta wave, coming to beta wave, it's a frequency of beta wave, it's 13 to 30 hertz, and it increases to above 50 at the mental state of it. When there is an intense mental activity, the frequencies goes beyond 50, parietal and the frontal regions of the scalp, and it be divided into 2 types, that is beta 1 and beta 2. Beta 1, it is the inhibited by the cerebral activity. So cerebral activity will be recorded by beta 1, and it is a mental activity like tension and other sorts of stress and everything will be detected by the beta 2. Coming to theta waves, the frequencies is about 4 to 8 hertz, and it is recorded as usual, it is recorded in the parietal and the temporal regions of the scalp, and it occurs during the emotional stress in some adults and also during the disappointment and frustration. Theta waves occurs, and whereas the delta waves are frequency is very less, it is 0.5 to 4 hertz. It occurs only for 2 to 3 seconds. It mainly at the deep sleep in the premature babies and even in this serious infections, organic brain diseases. This is the ECG wave pattern. Delta wave will be like this and exhibits high frequency. So the wave patterns are like very shorter, and whereas alpha wave is like this, and it's all about an irregular patterns. There is no proper pattern, like it is just an example of it. Now next is the placement of electrodes. This is the placement of electrodes. Usually for EEG we use 10 to 20 electrode systems. It is devised by International Frederick of Society of EEG. This is the pattern of placements of electrodes on a scalp. We have an arseon to inion. It is nothing but is the from the nose point, from the nose forehead, we will have a nose as a point, and then it will be go back to the back side of the skull. This is the center point, which is a midline. This is the midline, and F is Z, C is Z, P is Z is nothing but is the frontal area, this is the central portion and this is the parietal region, and FP1 is, and F is the frontal and T is the temporal, P is the parietal. So, this is the thing, and it is a midline, and half of the it is left hemisphere and the right hemisphere. The odd numbers will be placed in the left hemisphere and the even numbers electrodes will be placed in the right hemisphere, and this is portion is the frontal area, and here we will place the temporal electrodes, and this is the parietal is the back side of the head, and this is the central portion of the scalp. And this is the auricle electrodes are placed in the A1 and A2 position near the ear lobes, and this is the setting as after the placing of the electrodes, the brain will capture the electrical activity. This is the recording setup. So, we saw, right, electrical electrodes, the placement of electrodes. After the placement of electrodes, these electrodes are being connected to the jack box, and this jack box, what does the role of the jack box is nothing but it all all the electrodes are connected to the jack box in a sequence manner, and then it is being given to the filters. The electrodes pick up the signals and then it is being given to the filters. Before that, there is a montage selector whether the electrodes, all the electrodes are connected in a proper manner and it is comparable, and then it is given to the filters. We have 3 filters, that is a high frequency low pass filter, high pass filter, low pass filter, high pass filter and the notch filter. Now, low pass filter filters the high frequency noises and the high pass filter filters the low noises, and then notch filter filters the interfacing currents, and then after the filters it moves on to the amplifier, because the brain waves are very small in amplitude, it is a weak waves. So, for amplification we are going for an amplifier, and then it is the amplitude is amplified, and then it is given to the pen or paper ECG. Before, in the older periods, we were using the pen and the paper ECG. Directly after amplification it will be drawn, and after that nowadays we have a AD converter, A to D converter, where it stores the signals and then it is being stored in the database, and then using digital filtering, the sensitivity of the waves can be improved. So, that digital filtering also done, and then finally it is being displayed on the oscilloscope. This is all about the recording setup of the electrodes. Theory portion, as we told, as I described, this is a description of the block diagram. So, coming up to the summary of this presentation, first we saw about the nervous system. What is the nervous system? It is the complex, highly complex organs which is the main portion of our body, which involves controls and coordinates all the functions of the body with the chemical signaling, and then it has a specialized cell called neurons, and with that neurons only all the functions takes place. And next we saw about the nervous system disorder. If there is any problem with the nervous system, we have a dysfunction, so that deals to the nervous system disorder. So, in order to find out the nervous system, we have certain examinations and diagnostic procedures, and then we saw about the basic diagnostics procedures is the ECG, EEG. EEG is nothing but is the electroencephalogram, which records the electrical activity of the brain, and then we saw about the placement of the electrodes, the 10-20 lead electrode systems, and then we saw about the recording setup of the EEG, and then finally reference. Thank you.